Today I'm going to share something about the mixture effect. I would like to say it's more like discussions rather than any confident solutions. So anyway, the mixture effect is very, very important. I would like to start my story from the epidemiology and toxicology. So if we want to find any environmental factor that can be correlated with some disease, usually there are two ways. One is epidemiology way, so you can do the statistical analysis, but sometimes we have been criticized for the causal effect. So this is the examples I got from my pharmacology study when I studied in Duke University. So one time they showed me something published on New England Journal of Medicine. So they find the chocolate consumptions and also the Nobel Prize winners, if you do the correlation analysis, they are very good p-values, very good. So here we know that the correlation sometimes doesn't mean causal effect. Myself as a toxicologist, I have been rejected by many journals in the past. They always say, "Dear Professor Fang, we apologize, we cannot accept your paper because of the high dose you use this time." So for toxicology, we know that environmental relevant dose is very important. So for me, I have keeping thinking about this all the time. Really, we think it's time to really mix them together in some good ways. So how to solve the problem I have just said? Actually, the exposome, I think, is a very good field we can use. I think we have learned a lot about the concept that we will not talk about today, but for the exposome, we have lots of challenges. First of all, we have a large number of chemicals and very low concentrations, and some of them will undergo biotransformation in our body. And last, we will have very complex mixture toxicities. So this is a story, it was published very early in 2002, so it's about something from nothing stories. That means you have 8 estrogenic compounds. If you use them for each individual assay, actually you don't have any results from them. But if you combine all together, you're going to have very good mixture effect. And this mixture effect can be explained by the concentration addition methods. So also I want to start my own story when I studied with Dr. Harry Streppin in Duke University many years ago. I have found something which is very interesting. The dust samples, this is real environmental mixtures. So when we use extracts from the dust extracts with those 33 cells, actually we find there are lots of adipogenesis from the extracts. Wow, this is very interesting to me. I decided to take this as my PhD project. And the effect actually is very strong. It's very close to the type 2 diabetes drug, right? Rosiglitazone. So we think this is very interesting. So after that, we really want to figure out what is the story behind that. So we think the PPR gamma might be a possible mechanism to explain this adipogenesis. So we have tried different type of dust, okay, and we get lots of positive information from the dust from different places. So it's very exciting. So we want to figure out what the chemical inside is causing the effect. It's making me very excited, okay? So we, as a chemist, actually, we want to use something called the fraction, direct analysis, which is effect direct analysis. Means we try to fractionate your complex mixtures into different fractions based on their different affinity with the chromatography. So we have different fractions, and we have identified a few fractions that have very potent activity from them. And after that, we have done a non-target analysis of these active fractions. Okay, so here we see very nice 9 peaks. So for 9 peaks, we identify the first 7 as fatty acids. The last 2, one is a frame retardant, the other one is like the plasticizer. I was extremely excited. I emailed this result to my supervisor at very late evening. Okay, I say I have found something inside, and one of the chemical is a TDCBP and a TBEP. I'm so excited. Okay. And on the next day, I tried these 2 chemicals for my bioassay. There's no activity at all. So it's not them. And then I thought maybe these are the fatty acids that's causing the effect. Okay? And I do the bioassay test. Yes, they are. So this makes me think a lot. Okay? It's very interesting. I find something that's not environmental chemical, but some endogenous things. Actually, it's from the spray of the oil during the cooking that cause the effect. But this tells me something. Okay, this method is very effective. But sometimes, you have big limitation. If you do the real sample can be really varied between different places. So this method can be applied for some specific locations, but if you want to apply this for the policy and regulation levels, it doesn't work at all. So when I start my own lab, I'm still quite crazy about the mixture effect. So I asked my students to do some mixture, experiments, use like 13 different type of chemicals here. So for the certain chemicals, actually we have found there are lots of chemicals that have the interaction with each other, like agonistic effect or antagonistic effect. It's pretty clear. But I don't want to go to the story here today because I have got lots of complaints from my students. They said they don't want to do the experiment anymore. The reason they give me is, so here you ask me to do the 13 compounds. So you are going to have 13 times 12, which is about 156 pairs. So each of them you ask me to 4 different type of ratios. And for them you ask me to do 5 different concentrations and 5 replicates. So you're going to have like 162 wells. And you ask me to repeat the experiment twice. Okay? So this means about 3 more hundreds plus for just such a mixture effect. Originally I just say I don't agree with you. This is science. We have to do it. But finally I agree with them because it's like 300 plates. They play with it to get like only 13 chemicals. So I find that this is very hard. So I keep thinking about this all the time. So the final destination is, can we study the mixtures of chemical cocktails not by 2 at the same time? So how can we simplify the mixture design? So this is very important. So I think there are 3 key questions inside. One thing is how to select the exposure chemicals. The other one is how to design their dosing concentrations, and then how to study the multiple exposure and multiple targets. So I think this is the 3 major targets for the mixture effect for chemicals. So how to start? I think we have to start from the chemicals. So we know for exposome there's just too many chemicals we can focus. So we have to shrink them down based on their risk. So I use the baseline risk, means we have to consider their exposures and also the toxicity in some way to shrink them down to something we can handle. So here we have developed our database, which is a human exposome database. Basically for this database we use EPA, the SIM3 models to consider the exposure level of different type of chemicals. At the same time, we also bring like the toxicity. I mean here we don't, because toxicity can be anything. So we just use predicted toxicities. At the same time, we have bring the toxify data from the experiments to consider them in these risk assessments. So from this one, actually, we have generated a user interface, so you can really look up any chemical with their predicted risk. So here, I just show you some examples. What type of chemical that can be important based on the risk. There are something like the dioxins. Probably they are very toxic, but their exposure level is very low. So generally, they may be not that high in the ranking. But here, I will not go to the details. But you can see there are something we can find from this risk. Another thing is we can go from is epidemiology studies. I think epidemiology is very powerful. I'm not an epidemiologist, but I think it's super helpful. So there's a paper published on Science about 2 years ago. So I think some of you probably have seen this before. So from this one, actually, they have some cohort studies. From cohort studies, they analyze the chemicals. Finally, they do the statistical analysis. So what type of chemical can be epidemiology correlate with the disease? And after that, actually, they create a mixtures. And with these mixtures, they do some biological study to study what is mechanistic for the toxicities. So they have found something. So here is a very good combination of the epidemiology and mixtures and also the toxicities. So I think it set up a very good model for the mixtures. And another one actually is a meta-analysis. Sometimes actually we don't have the cohort study because it's very expensive, but we have something we can use before which have been published. So the meta-analysis is very important. So here I ask my students to do something like EDCs and the semen qualities. Actually we have reviewed over like 1,000 papers. Finally, there are only like 7 of them have popped up with positive during different type of studies. So I think through this way you can find some chemicals that can be epidemiologically correlated with the toxicity we are interested, especially the health outcome we are interested in. And also we can use like the enhanced cohort databases. It's a cross section, but you can also do the statistical analysis to tell you what type of chemical can be related with adverse health effects. And another thing we still want to go to is a toxicology network. I think this is very important. So based on what we have studied on the toxicity of the chemicals, can we predict what type of chemical can be linked to some disease? So how to do this in a more high throughput way? So here we have to get lots of databases. So database can be the like the CTD databases. I met the Caroline yesterday. I think it's very useful databases. At the same time, we also curated other databases which have been published online. So from all these databases, we think we can create how one chemical can affect the biological targets of one chemical. And then we have the disease databases, how one chemical can be linked to some particles, some targets. Targets can be linked to some disease. Through this way, actually, we have created an online web called exposomics.com. So from this website, actually, you can do, you can input the information of your chemicals, or you can input your disease information. For example, I have the red one here is the disease. We can, through this type of high screening methods, we can know what type of chemicals that can be linked to this disease from the toxicology, network through the PPI interaction, protein-protein interactions. At the same time, you can also see how the chemical they can interact with each other by some specific proteins. So through this way, we can use the toxicology network to give us some idea what type of chemical can be linked to the disease. So for the dose concentrations, it's very difficult. For the first dose concentration, I say the data sharing is very important. Enhance have done very good job for us. They have opened all the data for us. We can look up any concentration information we know. Sometimes we can also use the references, that can help you determine what is the human relevant concentrations. Sometimes, but this is not enough, okay? Because lots of chemicals that cannot be quantified because you don't have the standards or you don't have the methods. So it's very difficult. Another thing, I think we can use reference dose. So this something has been proposed in the past, but it's a little bit high concentrated, but it can still be used for some safety values. Another thing actually is the prediction models. Most of them actually is based on the PBPK models, like the IVV models developed by the by the US. That's very powerful to predict the concentration of those chemicals. So here I also say we can use some AI models to predict the endogenous concentration of the exposure. So this is a paper we have published last year on human exposome predict models. So for these models, we know the hypothesis I got is we have made the concentration of like about 200 compounds in the human blood. So for example, we know the concentration of the BPA, but we don't know the concentration of the BPAs. But BPA and BPAs, they share lots of similarities for the exposure routes and for the xenobiotic transformations. So if we can got all the parameters that can be linked to the steady state concentration in the blood, for example, half-life predictions, and if we have the SIM3, which is the external exposure levels, and we have the distribution volumes, we got these parameters into the model, so we can set up some prediction models to predict the concentration of many compounds in the blood. So this is the prediction power. We use the random forest models for this, optimization, and we found actually the prediction power is quite good. And use these prediction tools. Actually, we have predicted the concentration about 8,000 different chemicals in the blood. So if you see the accuracy of them is within 1 order of magnitude. So for the analytical things, maybe we think it's already quite huge. But for the exposure, if you think about the variabilities during the daytime and the evening time and also different days, different people, I think this concentration can at least be used as a reference for our experimental design. So finally, if you want to study the multiple exposure and multiple targets, actually it's very difficult. But now you are not studying one chemical, you study multiple chemicals. So because I was trained as an omics people, so I think we still use the different type of system biology methods to figure out how the chemical mixtures can affect some targets. And if you know some target, and later we can use some type of methods like the protein pull down assays, also some type of the computer assisted methods like the molecular docking dynamics or some AI models. And this the toxicologists have lots of models you can use. We can predict whether one chemical can be correlated with activity of some certain protein targets. So it's very useful. At the same time, I would say for the mixture effect, we can never forget the mass models. Like the mixture model is quite important. So here I will give you some example later how to do this. So this is something we have done a few years ago. So here I'm quite interested in the EDCs. So we have collected the concentration about the 23 EDCs from the previous blood or the urine samples. So from this one, I have designed the series of dilution in this way. We can use the geomean concentrations, which is very human relevant. But at the same time, we know the geomean concentration is not really like the worst scenarios. So we can use the 2 geomeans, 5 times geomeans, and we can also use the max as a worst scenario for the exposures. So here I use MSC cells, which is very simple endocrine disruptor good cell models. So we use a different type of omics to see how this mix can affect the results. So here is some of the data I show you. So basically for lots of the metabolites, we see there are lots of changes. It's not very typical monotonic dose responses, but you can see there has been changed by even the human relevant concentrations. So this is quite encouraging. In the mixture, they are really making some impact. So in order to figure out, in these 23 mixtures, what are the causal compounds? What's the most important contributor in the mixtures? This takes about 1 year to figure out. There are a couple of ways. So what we do first is we try one compound by one compound using the human relevant concentration, but the result is terrible because if you use a low concentration, the phenotype is not stable because there are lots of probably combination effect from the different chemicals. And if the phenotype is not stable, you are going to lose your phenotype during the experiment. So it's very hard. So finally, we use a method called the count-out method. This count-out method means you have 23. I remove one chemical by one by one. And then I want to see for the phenotype I have observed which makes the biggest contribution. If I remove some compound and there's no phenotype compared with the control, means this compound is very important. So through this way, we have to really use a good analytical method. For example, I shrink the 1 hour metabolomics method to 10 minutes because we already know the target, so we can make the high throughput screening for these different targets. So through this way, actually, finally, we have found the two compounds. One is DHB, the other BPA, that play the most important roles in the mixtures. So for the details, I'll skip it today. And after that, actually, we have some unparched data. So I want to make more ambitious mixtures. So here I try about 200 chemical mixtures. So this one includes most of the common endocrine disruptors we have known from the previous studies. And we got their concentration from the cohort studies to make a super mixtures. And mixtures, we have divided them into two. One is the environmental pollutants, the other one is the food contaminants. Because you have like genistins, also like the isoflavins, we know that it can be as estrogenic compounds. So we also include them inside. So here this is a design we use. We based on the GM values, so like 1 GM, 15 times GM, 20, 30, 40 is different type of one. All these omics to figure out. So for the metabolomics field, we have about like 50 different type of metabolites. For the genome, we have seen lots of changes, starting from the 15 times GM, but I show you the 30 times or 50 times now. So definitely make some changes. So upon all the pathways has been disrupted, we find the CoA. The CoA pathway has been affected a lot. So here we have developed a specific CoA profiling methods to profiling all the possible CoA for the for the metabolic disruptions. So here we find definitely for the 4 mixture and also environmental mixtures, they definitely play a big role in changing this CoA metabolism. So finally, we try to combine the transcription data and also the metabolism data. Actually, we find there are 3 proteins that can be affected a lot. The first one is carnitine acetyltransferase, which has been inhibited, and the other 2 actually is the carnitine palmitoyltransferase transferase. So these 2 are very correlated with the carnitine metabolism, which is the lipid metabolism. So it shows us that if you use omics, you really can find something new for the mixtures. And if you have the mixtures, actually another thing is a model, and model is very important. So the BEQ model has been widely used to predict the mixtures. So how you do that? You know the concentration of chemical in the mixtures. So here you have the REP. REP is based on the dose response of a specific compound correlated with some strong reference chemicals. So you get some reference values for this one. So you have reference values, you can do the concentration addition. You will get calculated values from these chemical mixtures. So because time, I just show you something we have done for the dust a few years ago. So here we have curated the concentration of different type of pollutants in the dust. We want to see which type of chemical in these super mixtures they are really making the impact. So from this like 350 different type of chemicals in the dust, we do different type of assays based on the intoxicity data we have got used in the mixture models. So here I just give you an example for the AHR agonistic effects. So for this one we have found a few agonists, which is one of them is the pH samples, which is expected. Dioxin definitely is not inside because the level is too low. And here actually we find the most important contributor is a pesticide we use in our household. Just through this type of experiment, I tell you, sometimes we always follow the new compound or old compound. For the exposome, to me, I think the magic is we can put all the chemical at the same levels. We compare their contributions to see which one is more important. So in the future, if we find another new chemical, we know the concentration, we can just input these models. Tells you whether this compound is important in this toxicology endpoint. I think this is quite important. So use the data we have predicted from the human exposome predicted models. Actually, we have applied this for different type of assays too for the human blood levels. So here the result is also quite astonishing to us. We find that lots of environmental chemicals we really care. Actually, they're not really making the difference. There's a lot of like food additives. The food additive level is very high, and they actually can be more important than the environmental chemicals. But there are lots of uncertainties because it's just based on models. And also the high throughput screening is very, very important. So we have done this for the immune response. We have selected lots of chemicals. They are very important. So here we just use a mixture test. So we find actually there are two compounds inside, which is very important. So one is a PFOS. PFOS compound really can deactivate the LPS from bacteria, and this LPS bacteria can really affect the asthma. So due to the hygiene hypothesis for the immune protections. But here I also want to say that because you have many targets, so sometimes the computational things like the molecular docking and also the molecular dynamics and also the AI models can also help you to predict. So here I try to summarize, for the mixture study. This is not the final one, I think, for the mixture study, but something from my own thoughts. For the chemical selection, I think that we have to combine the epidemiology study and also the toxicology studies. So this one can give us lots of information, what type of chemical to select. And also baseline risk is very important. For the concentration, we can rely on the body monitoring, but it's not enough. We have to use prediction models for different type of chemicals, especially for large number of chemical predictions. So finally, for the toxicity mechanism, I think the omics is a very good way. But at the same time, you need to have lots of mathematical models to predict what's the mixtures. Finally, you also need some IT models to help you to find which compound might be more important. So finally, I would like to show my great appreciation to my students at NTU and also at Fudan University. Also, I want to show my special thanks to the data sharing from the NIHS, EPA, and other toxicology databases. This is super for us to design our experiment. I really appreciate it. So finally, I also appreciate Dr. Beate Ischer from Germany. She also gave me a lot of experience on the mixture models. So this is my talk. Thank you.